So as time has passed, the amount of information we have on expression experiments has increased exponentially. So as you can see, this is the amount of, in this graph, the amount of arrays we have in the gene expression omnibus, which is a publicly available resource containing expression experiments. And as of 2010, the last measurement on this graph, we have over almost 350,000 arrays publicly available. So if you actually take the current information, it's, I believe, over 700,000 arrays publicly available in the gene expression omnibus, which is essentially off the screen. So as the amount of expression data has increased, it's been increasingly valuable to develop techniques which take this scale of data and actually compare in the microarrays in order to get information extracted from them. So what we want to do with this project is to create a BLAST-like tool to actually compare microarrays across species. So many of you are probably familiar with BLAST. So BLAST is a tool, you take a sequence and in one species and you look for where are in other species sequences which are very similar. And so this is relatively easy for a sequence compared to expression data. So sequence, there's kind of a default code of AGCT, which is conserved across all species. In expression, things are much more complicated. Conditions can vary, and that results in data which, even though may be different, might be due to conditions and not due to a lack of similarity. So the technique we use to compare expression experiments is a machine learning technique called Kotraining. So the way this works is we take one array in each species. So let's say we're comparing human and mouse. So let's say we have, for the human array, we have an actual microarray, and we have some kind of text attached to the array. So if you look in these publicly available repositories, you'll have an abstract, for example, which will tell you, well, this was the experiment we did. We tested a change which occurs in diabetes, and this gives you some information about what actually occurred and what is in the microarray. So let's say you have this information for both of your species, for your arrays. And so now you actually want to see how similar are these arrays. So the first thing we can do is develop models to actually test the two individual pieces of information we have. So we have a model which compares the expression data. So the human actual raw data, the mouse raw data, and we have a second model which compares the texts. So the text from the human array, the text from the mouse array. So for both of these, we have a typical machine learning setup. We want to train a model, and then with our training, some set of training data, and then test the model on all of our remaining data. And once we do that, for each of the two models, we get a score. So let's say these two microarrays happen to be similar based on the data, the expression data. And let's say the microarrays are also similar on the abstract. There's similar words that suggest that there's some kind of textual similarity. So now we can actually use a technique called Cotraining. So what Cotraining allows us to do is combine these models in a way to improve their overall performance. So the core of this is what we do is have an iterative process where when you take the pairs, so let's say for this pair we're looking at, we see that it's similar on text. We see that it's similar on expression. And so since we know that text and expression really actually can contain, to some extent, orthogonal information, we think that this is a particularly likely pair to actually be similar. So even though we didn't necessarily start with a label on this pair, we didn't actually know if it was similar or not, we can kind of assume that this is a particularly good pair, and it is very likely to be similar. And so what we do is we actually take this pair, we determine that it's the same under both metrics, it's similar both times, and we actually use it as training data for another iteration of our algorithm. So what we do is we iterate. Each time we find pairs which are similar in both data sets, or excuse me, using both models, and then we iterate until we decide to stop. So let's talk about what the two models we use are. So the first model is to compare the actual raw expression data between species. And this model is derived from previous work in our lab. So first thing we have to do is figure out how to actually represent a microarray in a form that we can actually process. And so what we do is we build a permutation matrix. So we have a set of orthologs. These are genes that we expect to be present in both of the species that we're looking at. And for each of these orthologs, we have some kind of rank, which is for example, if you score all of the expression values in the array and you order them, what is the rank? What order is it in? So we use order, we use rank, and the reason we do that is because when you look at publicly available data, things have been normalized in many different ways. So if you have different methods of normalization, however, one of the things that tends to be preserved is the rank of the genes. So if something is the most expressed, the normalization may change the value of that expression. It's not actually going to say, no longer is the most expressed gene, it's now the second most. So rank is useful to broaden the use of the algorithm. So additionally, we restrict, when we look at genes, we restrict ourselves to orthologs. So orthologs are genes which we expect to have similar function in human as well as or whatever two species we're looking at. And we define those orthologs from, or we pull those orthologs from some database. We actually use them Paranoid. So once we've built a permutation matrix of these orthologs by their ranks, we actually have to figure out what kind of distance metric can we use to compare them. And so this is what we do. And the important thing to take away from this is just that it's the L2 distance of the difference of two microarrays, one from each species. Let's say that's a human array and that's a mouse array. And then it's multiplied by some weight. So what this does is essentially it's a weighted score based on all of the differences between the two arrays. So what does this weight actually kind of encompass? So the weight tells us not information about in particular orthologs, but about in particular ranks. So what do I mean? So let's say you have a gene in human, which is the 10th most expressed gene. And you look at that same, the orthologous gene in mouse, and let's say it's the 50 most expressed gene. So maybe that seems like a big difference. Now let's say it's the 500th most expressed gene in human, and the 550th most expressed gene in mouse. Now that's a larger absolute difference, but the actual rank that we began with was much lower. And we expect that that's probably important. And so the weight actually takes that into account. So once we've learned a weight, we actually have this metric here, which allows us to take any two arrays between our species and get a score telling us how similar or how different they are. So after we have a score for the expression arrays, what we want to do is also develop a score for the text attached to the arrays. So the technique we use to do that is latent semantic analysis. And for those of you with a more machine learning bent, latent semantic analysis is essentially principal component analysis on a bag of words. So how do we take a raw abstract and actually put it in a form that we can actually do PCA on it? What we do first is take all the words and stem them. So stemming is a process of just removing the suffixes. So if you have expression, expresses, all of these things, the word of interest here is express. And so we just pull the stem out and keep that. Additionally, we have a blacklist of words that don't have content. So words like a and the have useful semantic meaning when you're considering a sentence in order. But when you have a bag of words and words no longer are consecutive in how you represent them, these words actually don't really help you understand the documents. And so we remove all these kind of connective words. So once we've removed a bunch of these words, we've stemmed them, we build the actual bag of words. And so what a bag of words is essentially is just an array again. And it's an array of words by documents. So each entry in the array is the number of times word X occurs in document Y. So once we've built this bag of words, there's a transform we perform called the TFIF, or Term Frequency Inverse Document Frequency Transform. And what this does is it allows us to kind of pull out the particularly interesting relationships that we can see in the text. So what does this do? So there are two components. The first is term frequency. So all this does is it weights words based on how often they occur in the abstract. So for example, let's say the word diabetes shows up 10 times in one piece of text. Well, we expect that that's really important to understanding that text, especially compared to another abstract where the word diabetes, say, only shows up once. So that's what the term frequency transform does. The inverse document frequency is kind of something similar, but from the opposite direction. So imagine the word expression, since we're looking at microarrays, probably shows up in hundreds of the different abstracts, maybe even every abstract. So we expect that the word expression is probably not very good for differentiating between arrays. But compare this to the word diabetes. Diabetes may only show up in 4 or 5 abstracts, and thus that suggests that it's much more useful in actually telling abstracts apart. And so that's the inverse document frequency transform. Okay, so now we have a bag of words, we've transformed it. What we want to do is actually perform the PCA step and to essentially perform dimensionality reduction. So what this does is gives us a reduced dimensionality reduction, or excuse me, a reduced dimensionality representation of our data. And the use of this is that when you look at the full matrix, there's a whole bunch of information which contains information about every document and every word. And this is probably extraneous and contains a bunch of noise and information that's just not useful for comparing documents. And when you reduce the number of dimensions that you look at, you essentially throw out a bunch of the information which does not seem to capture the difference between documents very well. And so once we reduce the number of dimensions, we can use this matrix to actually compare any two abstracts. And so how do we determine how many dimensions to use? We actually perform training to figure that out. Okay, so just to review, so we have a whole bunch of different abstracts, excuse me, a whole bunch of different experiments from each species. For each pair of one experiment from human and one experiment from mouse, we get two different metrics. We get a metric on the expression data, and we get a metric on the abstracts. These each give us a score of how similar or how different they are. We take pairs which are similarly scored among both of the models, so let's say both similar, and we use them as training data for our next iteration of the algorithm. And we continue to iterate until we've reached a stopping point. Okay, so how do we actually test this? So we extracted all of the human and all of the mouse GDSs from GEO, of the gene expression omnibus. So GDSs are essentially data sets, and they're the well-annotated data sets in the gene expression omnibus. So the reason for using the well-annotated data sets, of course, is that we can do a better job figuring out if we're actually correct or not when we've classified something based on all of the metadata which is actually attached to these arrays. So the number of data of actual arrays we're looking at is about 7,000, 3,700 in human, and 3,100 in mouse. So our initial training data, we actually begin with a set of hand-labeled pairs. An expert pathologist labeled 138 pairs, and that was our initial training data. And we take the orthologs we use from an existing database, which is in Paranoid. So the first experiment we did was cross-validation. So we have 138 training examples. So let's take out about 10% of them, 13, and train on 100 some of them and test on the remaining 13. And so what we did is we did cross-validation and we compared two techniques. So the blue line is the co-training technique that I just talked about. And the green line is if we just take model one, we just take that expression model and use only that. And so there are two important things to note. The first is that as you go and continue training for multiple iterations, you do see an increase in the cross-validation score, which is good. This suggests that you are, in fact, doing some kind of reasonable synchronization of your two methods when you are training. So eventually, you do get some amount of drop-off, and this is probably due to overfitting. You additionally see that the co-training technique in general does substantially better than the expression data alone. That being said, we expect the co-training technique to do better than the expression data alone. And the reason is that when we have those hand-annotated pairs, part of the reason we determined that they are similar, of course, is that the texts are similar. So there's a little bit of kind of causality problems there. That being said, the important thing to take away from the cross-validation is that you do see an increase in success as you have multiple iterations in training. So we did other tests in order to actually verify that the fact that we see that the training is occurring is actually giving us some kind of interesting result. So one of these statistical tests we did is basically, again, train the model through multiple iterations, and then keep track of how large the overlap set is between our two models. So what is the overlap set? That's essentially experiments which are similar in human in both models, or excuse me, experiments which are similar between the two species in both models. So as the number of iterations increases, as we see a similar pattern to the cross-validation results, we see a peak around iterations 6 or 7, and that's when we see the most pairs which are considered to be similar in both models. So now that we've actually done this method and we have a set of pairs that we think are similar between human and mouse, we can actually do another form of analysis, which is attempt to find genes which are correlated between the two species. So imagine you have these hundreds of similar microarray pairs, and you take a set of human genes, and you can ask the question, well, what are the mouse genes that are most correlated in all of these similar experiments? And so that's what we did. And to begin with, we used a set of cell cycle genes. So cell cycle genes are fairly well studied. So basically, we began with a set of human cell cycle genes. We ended up with a set of mouse genes. And then we performed Go enrichment to determine what terms showed up with those mouse genes that we found. Unsurprisingly, we found lots and lots of cell cycle related terms. So again, we compared the co-training technique, which in the red, versus the expression technique alone in the yellow. And for virtually all of the cell cycle related Go terms, you do see a greater enrichment in the co-training technique. So that suggests, again, that the co-training is actually doing a better job in finding similar microarray pairs. So the previous set, the Go cycle terms, or the cell cycle terms, are things that are fairly well studied. Well, you can also do kind of a query on things that you don't know as well. So imagine you take the genes in human that are known to be involved in type 2 diabetes. Well, there's no real good mouse model which is thought to have a similar expression pattern to humans with type 2 diabetes. So perhaps our technique could help us actually identify genes which could be important in mouse that actually have such a relationship. So what we did again is we took human genes that are related to type 2 diabetes, and we found the set of mouse genes that are most correlated. First thing we did is we looked at the Go enrichment, and we found enrichment for terms such as metabolism, biosynthesis, and immune system, all these things which are known and expected to be related to diabetes. Additionally, some of the mouse genes which show up are genes which are known in humans to have involvement in type 2 diabetes. For example, IL-18 is known, polymorphisms of IL-18 are related to insulin resistance. And versions of IKB in human have polymorphisms that are known to be associated with diabetes. So, in conclusion, we developed a co-training technique that allows us to compare expression experiments across species. So we do this by incorporating a new dimension of data, textual information, to supplement the expression data that you begin with and most other techniques use. So the technique allows you to additionally train your model with just the expression with additional good labeled pairs that you find by synchrony between the two methods. All right, so I'd like to acknowledge, in particular, Hai-San Lei, who wrote who wrote the paper on which the first model is based. And thank you. Do you have any questions?
Hi, thanks. That was interesting. Can you clarify for me where the text comes from? Is it from the descriptions in Geo? I wasn't. Mm-hmm. I'm sorry if I missed it.
Okay, so GDS datasets in Geo have abstracts attached. Now, so each of these abstracts is attached to the experiment as a whole. So not a specific microarray, but a set of perhaps all of the microarrays in the experiment.
Okay, so those are abstracts of papers, presumably, that are associated with those data sets.
So that's presumably, yes. Of course, people can put as their abstract. It's not necessarily directly correspondent to some published paper.
Okay, and now to an actual question. Sorry, that was just me being a sleepy, I think. Can you tell me whether you thought about comparing, when you did your gene expression, your Go term analysis, did you think about comparing co-annotation of those genes? So, of course, we have lots of annotation to gene ontology terms. So, did you compare to what's already been annotated to those gene ontology terms? So whether, like, the mouse genes and the human genes were both annotated to the same gene ontology term that you're picking out?
Right. So you're saying in a little more coarse grain, or fine grain, for some gene known to have one specific Go term, if the mouse gene is corresponding to it had the same Go term.
Right.
Right. So we did not do that specifically, but it definitely can be done.
Yeah, I think that would be an interesting comparison to make, whether you're picking up something beyond you can already get with the annotations that are out there. Thanks.
That was very nice. You showed the results for expression only and expression in text. How about text only?
Mm-hmm. Right, so as I mentioned in the last question, so the text sometimes corresponds to multiple individual arrays. So while we can compare one microarray to another microarray with the expression values, of course, the text actually gives us a many-to-many mapping. Because you might have one abstract which corresponds to 4 or 5 different arrays, and another abstract which corresponds to 4 or 5. So you can't get the same granularity of comparison. And so that's why we only use the expression technique.
Right,
so
but you could subsample and actually test, right? I mean,
right,
okay.
that's true, but one of the things is you might find, for example, control and experimental conditions in that set, right? So you have an abstract which corresponds to both the control array and the experimental array. So you could, for example, just be comparing 2 control arrays if you sample randomly from
Right. Okay. I see.
those
Thank
2
you.
experiments.
Right.
Thanks. All right, thank you very much.